Hey everyone, welcome to RegenMed Global, the podcast, where we bring you the newest breakthroughs in medicine and research to help bring hope in fighting incurable diseases. My name is Cheyenne. Today, we're here to discuss a major breakthrough that holds promise to a potential cure to diabetes. Leading this research and speaking with us about this, we have Associate Professor of Regenerative Medicine at Weill Cornell Medical College, Dr. Zhou. Dr. Zhao, it's a pleasure to go ahead and have you. Do you mind maybe just starting off by telling us a little bit about your background and what you do?
Cheyenne, thank you. It was a great pleasure to chat with you. I have a PhD in neuroscience, in fact, almost 20 years ago. But after which, I decided that regenerative medicine is the future. So I decided to work on regenerative medicine, and eventually we went on the path of um, trying to find new treatment, new regenerative medicine treatment for diabetes, which is a huge disease. We have almost 40 million patients in America, over now, 40 million, increasing every year, diabetes in America alone, and over 500 million around the world, by the way. So it's a tremendous disease, and we're trying to find new ways to treat them. That's been our mission.
Wow, that is awesome, doctor. Now, I mean, you've been doing a lot of work in terms of regenerative medicine, diabetes, and most recently, you did a preclinical study about, like I said, finding a potential cure. Can you give us a brief summary of that study?
Yes. So, so a quick summary before I give you a little bit background on this study. So the real thrust of the study is to develop a new autologous cell therapy to treat the most severe form of the diabetic patients. Okay, so these are the patients that require daily and often insulin injections. So there are about two populations. In America, it's estimated that there are about 4 million patients require almost daily insulin injection. This includes the severe type 2, so-called type 2 diabetes patients. So these actually includes most of the diabetic patients that we see them, and there are different severity of the diabetes. Most of them fall into the so-called type 2 diabetes. And then we have a smaller population, but still number in the millions of type 1 diabetes. So, and these are heavy users, and these the cell therapy that we devised that can treat this population of patients. So because of cell therapy, I want to just very briefly backtrack and talk a little bit about the cells that make insulin in the body and how we recreate these cells to treat the patients. So insulin is made by only one cell type in the body. These are the so-called beta cells in the pancreas. So each person has about 1 billion of these cells, and they aggregate into little clumps that together with other hormone secreting cells. So it looks like a ball of cells, maybe a few thousand together. And we have about 1 million of these clusters of cells. So these are the islets in the pancreas. So this is the only source of the insulin in the body. Not only that, an important point I want to make is that these beta cells are also the most exquisite glucose sensors in the body, right? So your glucose depends on your activity. You ate a cookie, you ate a chicken, or you run exercise. Your blood glucose constantly going up and down. And at night, during the day, there's also different rhythm, right? Circadian rhythm. So the beta cells can sense the up and down of the glucose and pump out just the right amount of insulin to control your blood glucose level within the normal range. So this is very important. When you lose the beta cells, when you lose enough of these, like in the severe type 2 or in the type 1 diabetes, millions of patients, now they can't make enough insulin. And then you need some extra sources. So normally, the way to treat these insulin-dependent diabetic patients is to load up a syringe with the insulin and inject yourself right before the meals. And in addition, you had to basically, for anyone who has treated themselves, you had to prick your finger with a real sharp object to draw the blood and measure your blood glucose. So it's very difficult to manage, and even for adults. And there are millions of patients, type 1 diabetes especially, have onset in childhood. So we're talking about a lot of children. It's very difficult to manage this disease. I should also mention that as we just chatted before, that the insulin was actually discovered 102 years ago in 1921. And at that time, how do they treat diabetic patients? They extract insulin from some animals. Back then, they cannot make it, but put them in a syringe and inject yourself. So 100 years later, majority of the patients still use the same method as 100 years ago to treat themselves. The problem is when you do a bolus insulin injection, the up and down of the insulin amount does not correlate with up and down of your blood sugar level. So your blood glucose still goes like this, zig and zag. It doesn't fall into the normal range. That's why for the chronic and severe diabetes patients, your glucose control, even using insulin, is not very good. So over long term, you develop all kinds of complications. I'll just give you an example. For example, for type 1 diabetes patients, onset in children, most severe, has almost no insulin in their body, and their lifespan is about 10 years shorter than normal healthy people. So it's a severe, there's a really strong unmet clinical needs to treat these patients.
Well, so how effective are I mean, I know that there's treatment, and it shortens the lifespan, but ultimately in terms of the quality of life, right? How effective are today's treatments?
Quality of life is very poor because unfortunately, the longer it goes, it's a chronic condition. Because if you don't have beta cells, if you don't have insulin, and you have to inject yourself for the rest of your lives, and you could miss a dose today, you could, things accumulate. So often over time, you develop all kinds of complications because of this fluctuation of glucose. There are a lot of complications. For example, neuropathy. There's your eyes could fail, your fingers start to tingle, your kidney have issues. I in fact have a relative that have severe diabetes, and he unfortunately passed a pretty young age because of complications. So this is not uncommon. It's a lot of burden. Measuring glucose, treat yourself, is a huge amount of burden. That's what we're talking about. And that's why us, among other people, are thinking, no, you lost your beta cells. How about we make new and more beta cells and do transplantation, right? Give your body back to what is missing. And not only you can secrete, get your insulin back, you also get the ability of glucose responsiveness. That is, you don't have to prick your finger to measure glucose anymore, blood glucose anymore. That these cells can just measure the blood glucose to pump out the right amount of insulin. So that will be, a great therapy. So that's what this preclinical study is all about. How can we achieve that? Not only that, can we do that in an autologous manner, right? So autologous, basically using your own body's tissue, fully compatible with yourself, to cure your own disease, versus a so-called allogenic. Now think about organ transplantation. That would be allogenic. Someone else or organ donor to give you a donation of the organ. For example, eyelids, or maybe the kidney, maybe a piece of the liver. That's someone else. So the immune system of different people are not really compatible. So if you receive a tissue from someone else, you have to apply immunosuppression to control the rejection, right? So, but if you can use your own body's tissue to cure yourself, that would be autologous. Now there wouldn't be rejection.
Okay. How impactful are kind of the therapeutic target of utilizing beta cells compared to, are there other therapeutic targets, or would beta cells kind of be like the primary one, or how significant would that be?
Beta cell is the key to this severe form of diabetes. There are other less severe Diabetics, you could use other drugs to control them. There are many drugs out there, metformin, others that many people use. But once you progress, your disease, diabetes, progress to a more severe form, I think me and many other colleagues believe that transplantation will be the best way to potentially cure the disease.
Okay.
So maybe I can launch into describing a little bit how we do that.
Yeah, let's go straight into that. I'd love to hear kind of
Yeah.
the next part of that journey of the study.
Yeah. So here is how we make new beta cells for autologous rescue, if you will, treating yourself. So we developed over the years, over the course of about 5 years, the study itself going back even longer, but this particular study lasted about 5 years, where we discovered that you can capture a tiny bit of your stomach lining, so using endoscope. I don't know whether anyone has done a scoping of procedure, but there are millions of people do this. Basically, put you to sleep. They put a scope, tube down your throat and look around your stomach and just to see whether everything is okay. I've done that once. It's very safe. It's done on millions of people. But during the process, if we find something, often they take a tiny bit of it. It's very small. It's like pinhead sized. And remove the tissue for examination. And the beautiful thing about the stomach is it has a lot of the stomach stem cells. So it's constantly recreating the mucosal lining of the stomach. Okay, so these cells are mostly like mucus secreting, acid secreting cells. But what we discovered is that when we remove a tiny bit of this gastric mucosa with the stem cells inside, we can put them into a tissue culture dish and use special culture medium to promote the growth of the stem cells. So the stem cells grow out, this stomach stem cells, and we can amplify them to huge numbers, like billions of them. Then we figure out a method to now basically to coax the gastric stem cells to make insulin secreting islets that are glucose responsive, right? So these are not, the stomach never makes islets. Islets is only found in beta cells, found only in the pancreas. So we are basically making the stomach stem cells to produce something, a cell type that it normally doesn't make. But because it's taken from your own body, that becomes autologous. Now you could then take this and transplant to yourself, right? And then correct your diabetes, secrete insulin. So that's a broad scheme of what we figure out. It sounds very easy, but we overcome a lot of technical challenges in the 5 years time and made that possible.
The preclinical work, it was done on mice, correct? Can you tell us a
Yes.
little bit about kind of just the overall kind of study design and before and after?
So once we figure out how to make the human gastric stem cells to make these islet-like cells, we actually give them a special name just to differentiate what we made from the stomach from the real islets, the real beta cells from the pancreas. So we call them gastric insulin secreting cells,
Okay.
or genes, GINS cells, genes cells, or
GINS
genes
cells. Very good.
islets. Yeah, so that's what we call them. So, well, we made them. We know that in tests, in tissue culture, they secrete lots of insulin. They also respond to glucose challenge. For example, we can put, low glucose, high glucose, low glucose, high glucose, and then they should respond, right? At low glucose, they don't secrete. At high glucose, they secrete more. If you even higher glucose, they secrete even more. So, right? So they need to respond to glucose by adjusting their levels of insulin secretion. So we did all these tests in vitro. But the most important thing is, can we rescue a real diabetes in a real animal, right? In animal models, we can't do that in human yet. So what we did is we have a standard rodent model of diabetes. So the mice, and basically there is a chemical we can use that was done many years ago. People discovered that there's a chemical you can inject, and then that chemical very specifically ablates the beta cells in the pancreas. So it's almost like this severe form of diabetes. You lost your beta cells, you lost the insulin secretion, and then the glucose just skyrockets. It's very high. If you don't treat them, the mice eventually will die from high, high blood, very super high blood sugar level. I would say it turns into more like a maple syrup, right? It's crazy high. At this point, before the mice get sick or anything, now we transplant or genes edits, right, made from human. And okay, then you ask, well, how can you transplant human tissue into mice? Are they going to be rejected? Yes, but these mice are genetically engineered so that they will accept human donor tissues. So it's specifically made for these tests. So now we have a extremely diabetic mice. We transplant these genes islets into this mice. What happened is the next day, the high blood sugar level plummets to normal level.
Wow.
We were all amazed how fast it just goes straight down and become normalized. And then we just measure them every day. We measure them for many months, and it remain normal. And then we'll say, well, what if we remove the grafted genes edits? Because these are the ones that are secreting human insulin in mice and normalize their basic blood glucose. Now, the prediction is if we remove this human grafted tissue, and then there are no source of insulin left, right? So the glycemia is going to jump back. It's going to become very high level blood sugar level. That's exactly what happened. If you remove the graft, it goes rocket back again. So it's completely controlled by the grafted cells. So we were very happy, and you can do different physiological tests like glucose tolerance and things like that, and it just normalized. So we were very happy. The cells work very nicely.
Was it just like an injection, or what was there a different method of? Okay, it was an injection.
Yeah.
Yeah. And so when you guys collected these genes islets, right? So it's from a person's stomach, or how did you guys derive them?
Yeah. So first, let me elaborate a little bit about the injection. So in human studies, most of the time people take the islets, for example, donor. Sometimes people do small number of transplantation from donor islets, isolated from donors, and they normally infuse them into the liver, and that's somehow the clinicians figure out that's the good way to put the islets are. In animal studies, we normally don't put them into the liver, but put in other places where it has good blood supply. So there are different places you can situate them, and they all work, basically.
So where's some places that you would put them?
For example, sometimes so there's a membrane inside the stomach, it's called omentum membrane. People wrap them in there. Sometimes people put them inside the fat tissue because they also have a lot of blood vessels. Sometimes people actually put them under the so-called the kidney capsule. So the kidney, there's a membrane, so very tight membrane surrounding the kidney. It's almost like a glove on your hand. So it's called the kidney capsule. And sometimes people transplant tissues under there in mice, not in human, really. And the reason is because it has a lot of space. You can put some cells in there, and also it has very rich blood supply, so the transplanted tissue will survive and prosper. So that's sometimes people put them under the skin, but that's not very good because skin has a relatively poor blood supply, so the tissues normally don't survive very well.
From it being derived from the stomach, like how were you guys able to do that? Was it? I'm very curious.
Yeah, so so for some of the initial studies, we got autopsy donor tissues from tissue banks, and then we take tiny bits of biopsy-sized to do this experiment because we can get many little pieces to test our procedure, right? The experiment doesn't always work. Most of the time you fail. You have to retrack your steps and work out the best methodology. So initially we did that, and then we also received biopsy tissues. That is strictly because from live donor. Now that's very strictly regulated. You have to have consent, patient consent, institutional review. There's very complex process, and then you have to de-identify the donors, so their identity is completely hidden, and we don't know anything about the donors. We don't. When we receive the tissue, we don't know. We have a number. There's donor number one. That's all we know, and then we can work with the tissue in a way that's completely preserve the privacy of the donor, so we get the tissues like that to work with. So we are very thankful to the donors from tissue bank, from live donors, to enable our work. So without which we can't develop therapeutics.
Right. That's great. Just overall, we talked a little bit about just like the preclinical work, but ultimately, if you were to just conclude it in terms of just any major important findings or just any conclusion about it, what would you say is kind of the most that you got out of this whole preclinical study?
I think the most important study we did is to demonstrate that the transplantation of these genes, islets, can immediately, effectively suppress hyperglycemia and maintain a normal level for as long we track them, many months. I think this is the most powerful demonstration that this is going to work in the probably in a clinical setting. But there's a long pathway, we can talk about that, to go from here to real therapy. But
Right.
we have the most important proof of concept that this could work.
That's awesome. So what are the next steps in this?
Next step would be work and more work.
Tell us a little bit more about what that work consists
So
of.
tell me, yeah, so right now what we have is a prototype, a proof of concept, what we have. But real cell therapy takes many more steps to reach there, a lot of investment. And not only in the academic lab setting, we also need to engage with the biotech companies and big pharmaceutical companies, because human clinical trials is very, very expensive, and it's beyond, we're talking about hundreds of millions of dollars.
Mhm.
And that's only the big pharma has that resources to carry it on. But before we get there, we need to make sure that the technology is highly robust. We can always make it better, right? More consistent. Every patient, for example, I'll tell you a few things that we're working on right now. One is we are collecting more donors and make sure that every time we make the cell product, it's going to come out functional, and it's going to be have the curative power. So its consistency is very important. You can't make a product that says our technology, it says, okay, I make this product, it works beautiful, the next one doesn't work so well, the third one may be a failure. So that's not going to work. So we need to optimize technically, make sure it's highly consistent from one donor to the next donor. The other that we're working on is that because type 1 diabetes is autoimmune disease, that is, the immune system somehow aberrantly recognized the beta cells as foreign and attacked and destroyed them. Now, many years studies have led to a lot of insight, but we don't know exactly why and how this happened in this patient. We actually don't know the definitive answer to that. But nonetheless, there are ways to protect these beta cells. For example, we could potentially express some of the immune modulatory molecules on our gene cells, so that when we transplant them back into the type 1 diabetic patients, and the newly transplanted cells can resist autoimmunity, right? So they won't be killed and they will functional. So we're working on that part as well. because T1D, type 1 diabetes, is really, afflicts children, and they are often the most vulnerable population. So any cell therapy, the first target population will be type 1 diabetes patients.
What do you think the biggest obstacle is to be able to proceed with the next steps? Do you envision any obstacles you're like, look, this is something we're concerned about, or this is something that we think we're going to have a trouble overcoming?
Well, I would say the more resources we have, the faster we can move, right? Because we have to work on a lot of things, different optimizations and testing different people, different populations. So it's really about manpower and resources at this point. The more we have, the faster we can move it to the clinical trial stage. In terms of obstacles, I think it's very hard to predict. You had to When you get to the bridge, you cross it. You have to solve it. But the great thing about scientists, Xiaoyan, is that we are great problem solvers. We sit, we work at the bench. We sit in our office, read papers. Our daily job is solving problems. So we can solve any problem that comes our way.
In terms of the next phase of the study, I mean, prior to going to any kind of clinical approach, how long do you see that kind of taking in terms of time?
Depends on the resources we have. Unfortunately, at this stage, from a proof of concept to a like really clinical prototype that can go into people, that stage, the length of that stage is entirely depends on manpower and resources you have. In my estimation, now, if someone comes up and give me $5 million, I know scientific research is very expensive,
Right.
especially therapeutic development. But if you have, let's say, $5 million, we can probably develop a prototype that can go into person in 2-3 years time.
Wow. So you're saying if all the stars align,
Yeah,
earliest clinical
we
trials
have
will be available?
we can get to that stage. We can get to almost the clinical trial stage if you have enough resources. But if you ask any other people that are developing therapeutics, that's always the choke point,
Right.
because that that kind of money at this stage is very difficult to come by. So you got to have some believers in this to invest, and basically probably in a setting of a small biotech company to now really move that to build a prototype. That's what we need to do. That's what everyone has to go through. The drugs might be different. Could be small molecule drug for treating this disease, could be a gene therapy to treat that disease. Here is a cell therapy to treat diabetes. But that's the stage to move that from an academic lab to a real therapy. This is the critical stage and also most difficult. I can tell you that, because requires some amount of money, and whoever put forward the money have to take some risks, right?
Right.
So this is the world of venture capital and other small biotech building. And, we for anyone who wants to take their research from the lab to a real therapeutic, you have to go down that path.
Let's say this moves forward into the kind of the clinical environment. How does something like this? How would this look like in terms of just how it's used? Would it be used on like as a one-time thing? How would it be injected? I mean, do you have an idea of how that would look like?
Yeah. So here's what I believe is going to happen. So let's see, we have a patient that's going to take this therapeutic. So the first step is a endoscope procedure, right? You go endoscope, takes I don't know, hour, maybe less, and then you wake up, you go home. And now we take a tiny bit. The tissue is very small. It's pinhead size tissue. It's tiny. And then to make a product, the current estimate is because we had to amplify it, we have to induce it, we have to purify the product, so take multiple steps. When we get to a product, we think that's going to be in about 3 months of time, 3 months to 4 months of a time. But not only we have a injectable product, we can also bank more products, so that if you need that in the future, we have that banked, right? So then we take it and give it to the patients. Right now, the technology is to infuse them into the liver. So I think we are probably going to do that as a first step to infuse into the liver. And also, there are other people that are developing technologies to enable subcutaneous transplantation. Remember, I said subcutaneous normally is not preferred because it's poor blood vessel. But there are ways to potentially enrich for blood vessels and put the product right underneath your skin. I think that would be great, and because it's a lot easier, right? You just inject, and that's it, and you go home, and hopefully everything is good. But in the event that over time, I don't know, this product can probably last for years, and at some point it might start to degrade in quality and function, and by which time we can go back to our freezer and take out the same product that we made before that we banked and give the person a second infusion. So that's our vision of how this can be done, and we can do that for thousands and millions of patients. That's the hope, not only in type 1 diabetes, but also severe type 2 diabetes patients.
That's huge. I mean, something like this, was it something that as soon as it's available, healthcare, the insurance would cover it? Is that something you'd be aware of, or is it something that would be separate treatment where it's like a choice where they can choose if they want to do it without the support of insurance?
Well, we need the insurance support, right? Because these therapies eventually is going to cost quite a bit of money. Right now, you may have heard some of these therapies are very, very expensive. It's true. But we have to look at it from two different angles. First, if we can mass produce a therapeutic, the cost is going to decrease dramatically to a level that's more affordable. And also, importantly, we need to keep in mind that for diabetic patients and severe diabetic patients, and they have all these complications, right? Your eyes start to fail, your kidneys start to fail, your fingers and toes start to basically change. And it's very, the side effect, the long-term complication is very bad. So now, when you progress to that stage, you have to spend a lot of healthcare cost to treat these patients. The burden is enormous. So I think, eventually someone has to pay for the therapeutics, and the patients can only pay for some of them. So the insurance company has to be willing to pay, but I think they will, because the complications are very expensive.
Right.
And once you have them, they don't go away. So you have to treat them for the rest of their lives. So I think the economic calculation would be such that the insurance company would be waiting for some relatively what seems to be expensive treatment, but ultimately it saves a lot of resources.
For sure.
And quality of life. We are not even talking about the quality of life. That's
Right, of course.
which is far more important than calculating the dollar amount. But that's something we have to think about. There are different aspects, the economics of developing a therapy.
Right, exactly. Now, I mean, just with the work that you and your team have done, do you see this potentially opening up treatments for other diseases too?
Yeah, that's a great question. So I think the way that we are approaching this, which is to take autologous therapy, right? Basically use your own body's cell to make some other types of cells that are missing or diseased, and just replenish your own body. I think that's approach we should think harder about how this can be done. It will be autologous. Could it be just throw it out there? Let's see. We have this stomach stem cells. Okay, we made islets that can secrete insulin. Could we make liver cells, right? Because there are a lot of people with liver failure, and you have a lot of stem cells in the stomach. Could we take them and make some liver tissues and help with liver degeneration or loss of liver function? Those are the type of things we should requires. I would love for more people to think about how this can be done. In fact, there are some hints, in the liver field, that there are certain cells can regenerate liver from the ducts of the liver. So, you know, we need to not only to study them, but push towards a real world therapeutic. That's what we need to do.
Right.
Yeah.
No, that's great, Dr. Zhao. And we talked quite a bit about the preclinical potential opportunities it could open up and the future it can have, the impact it can have on the future. I'm curious, is there anything else that you like to add, or anything else that was really important to you on this journey, on the study that you want to share with the audience?
Well, I would say that scientific research is problem-solving process, and we chose the topic of most importance to address. We certainly need a new generation of young people to help us solve a lot of issues, medical diseases. There are still a lot of them out there. I would love for people to be interested, and also very importantly, the society support is very important, right? Biomedical researchers, as I said, is quite expensive, and a lot of these being supported by the government, like National Institute of Health, where a lot of people get where the grants from to study these, but also biotech companies to move these from labs from bench to bedside. That's what they say. So support is very important. And Cheyenne, I thank you very much because this is the portal to go from scientists like me and talk to the general audience. It's important for us. I would love to address any questions that you have, the audience that might have for me. Just email me directly. I'm happy to talk about this, and just so that we keep a dialogue open. The public knows what we are doing, and can give you some tips how to be a scientist if you like.
I love this. I would love to open up the communication between you and the audience. If anyone has questions, reach out. I'm definitely going to provide that connection, and very excited about a collaboration like that, and very excited with the work that you're doing, Dr. Zhao. And thank you for the impact that you're making in the future of medicine. This is exciting from someone like me, and I'm sure everyone that's watching is really excited about the work you're doing.
Thank you, Cheyenne. I appreciate your time.
Thank you.
I appreciate the channel of communication.
Of course. Thank you, Dr. Zhao.